Wonderful. So I'm Erica Barnell. I'm an MD PhD candidate from the Washington University School of Medicine. I completed my PhD thesis in Obi and Malachi's lab, and with BC, who is our clinical collaborator, we conducted a clinical trial to understand if we can use personalized ctDNA micropanels to predict clinical outcomes for patients with breast cancer. My disclosure, is it moving? It is. My disclosure is that I co-founded a company my first year of medical school, Genoscopy, which was actually able to sponsor a portion of this event tonight, which I'm very proud about. So, Chief Science Officer, and I have equity in the company. A little bit of background on triple-negative breast cancer. So, neoadjuvant systemic therapy is widely used in these patients. About 30 to 65% of patients with breast cancer who receive this systemic therapy will achieve a pathological complete remission. However, patients who don't achieve PCR have a median survival of only 18 to 28 months. And so this recurrence is likely attributable to the escape of resistant cells, which are not really detectable with current tools in routine clinical practice and present with the clinical signs of recurrence later in the course, after the molecular kind of components have been realized. We have been able to use liquid biopsy approaches to understand circulating tumor DNA, and this offers a very high promise for understanding real-time response to chemotherapy and predicting clinical response in advance of those kind of outcomes. So while ctDNA in peripheral blood has been used to predict prognosis and therapeutic response for triple-negative breast cancer patients, most of these approaches use large comprehensive panels where there are genes that are recurrently mutated across all breast cancers. And with the reduction of sequencing costs and turnaround time, we can actually integrate custom micro panels for tracking disease and predicting clinical outcomes. So that's what we tried to do in this study. We hypothesized that these custom micro panels could potentially improve the sensitivity related to measuring clinical outcomes for patients with breast cancer and potentially identify those individuals who were going to recur in advance of clinical symptoms. So the sample collection is outlined here. We obtained baseline tumors at T0 in advance of surgery or chemotherapy. And then subsequently, we obtained blood samples at baseline at T1, which was cycle 1, day 3 of chemotherapy, during the time of the patient's operation for clinical resection, and then at 3 to 6 months follow-ups when available, when the patients were showing up to the clinic. And finally, if the patient relapsed, we would also collect a blood sample if available. The first thing that we did was whole exome sequencing of the baseline tumor. This allowed us to identify 4 to 6 variants associated with the individual's tumor and build a custom micropanel that we could use subsequently on the blood samples to identify recurrence of these tumors in the blood. And then we employed these micropanels for every single blood sample that we obtained at baseline, at cycle 1, day 3, at surgery, and at those 3 to 6 month intervals until clinical relapse, if applicable. The outcomes that we were looking at was the variant allele frequency for each of those 4 to 6 variants that we had in the microarrays or the micropanels to understand if we had recurrence of that circulating tumor. Here are all the samples that we collected. The first column, that kind of blue and purple square, indicates the whole exome sequencing that we performed on all 50 patients that were enrolled in the clinical trial. That whole exome sequencing data was used to identify the micropanels, the 4 to 6 variants associated with each individual's tumor. And that was employed at T0 at baseline prior to surgery as well as chemotherapy. Then the custom amplicon panels were built, so we identified those variants and we employed those custom capture sequencing panels on all the outstanding blood samples that we collected for each of those 50 individuals. Unfortunately, you can see that we didn't have a complete data set with all 50 individuals. It was a little bit sparse for some individuals that were lost to follow-up, about half of them. But you can see that maybe 25 patients had blood samples at baseline, at cycle 1, day 3, at surgery, and then at a number of 3 to 6 month follow-ups. And then I put stars with an R to indicate patients that had recurrence of disease clinically. So those are patients where they showed up to the clinic and the clinician identified a recurrence of disease. There were 4 patients in that category that had recurrence of disease with a blood time point within the last 6 months, and I'll be talking about those shortly. So the whole exome sequencing was very reflective of what you would expect for triple-negative breast cancer. There was a median of 77 somatic variants that were identified, a range of 1 to almost 2,000 variants. There were some hypermutators in there. And then TP53 was identified in 86% of all patients, so 43 patients out of the 50 patients that we queried. And all those TP53 variants were included in our micropanels. In total, there were 208 variants across all 50 patients that were selected for the custom micropanel that we employed on the blood samples. So here's an example of our ability to use these micropanels to predict clinical or micro ctDNA recurrence in advance of clinical recurrence. So on the x-axis is the variant allele frequency. Each line represents one of the variants that was selected for the individual's tumor. And then on the x-axis represents the individual time point. So you can see for this patient, at baseline, at cycle 1, day 3, at surgery, and then for the first, 18 months, they had no presence of these variants in their blood. And then about 6 months in advance of clinical relapse, we show a spike in, 2 of the 3 variants that we identified in those tumors. And then subsequently, at the time of relapse, there's clear indication that this tumor has come back and has recurred. Similarly, this patient had circulating tumor DNA at the time of surgery. During cycle 1, day 3, it did drop at the time of surgery and was potentially free of disease for the first couple of months. But then again, in advance of clinical relapse, we kind of saw that spike up, and then there was clinical relapse for that patient eventually. Another patient, again, that had clinical relapse, showing that spike after surgery during the patient's kind of chemotherapy course, and then it drops back down to baseline and in advance of clinical relapse, demonstrating the recurrence of molecular disease in advance of clinical relapse. And finally, this is a patient who never actually achieved clinical remission. So we showed in the blood that the circulating tumor DNA was present at surgery at cycle 1, day 3, and never actually dropped to baseline, and they relapsed at about 2 months. So these are 4 patients where we were able to capture blood 6 months in advance of clinical relapse, and all 4 of them kind of showed this molecular increase in circulating tumor DNA in advance of that time. And then conversely, I just wanted to show the patients that we didn't see any molecular relapse. These are 4 representative examples of the 17 patients who did not show clinical relapse during the clinical trial. Most of the 17 looked exactly like this, where you actually just don't see any change in the variant allele frequency over time. And then the last thing that we wanted to do was associate our molecular signatures with the clinical response and demonstrate that pathological complete response was associated with a potential increase in our ctDNA signatures over time. And so these data show that of the 17 individuals that did not have pathological complete response at baseline, 47% showed a positive ctDNA ultimately during their clinical course. Conversely, of the 8 individuals with pathological complete response, only 1 patient showed a positive ctDNA during the timeline in which we followed them. So that indicates potential increase of our signature to predict a kind of PCR at the beginning. So in conclusion, we were able to build these custom micropanels very inexpensively and in a really good turnaround time to query patients with triple-negative breast cancer. These custom panels potentially provided improvements relative to traditional universal panels that have many genes and are universal to everyone, but not custom to the individual. We decided that and demonstrated that this ctDNA evaluation can occur at approximately 6-month intervals and still capture kind of recurrence in advance of clinical response. And finally, we did have a large dropout in this study, so larger studies are going to be needed to fully demonstrate that this kind of approach can work for these patients. I want to acknowledge the Griffith Lab, my clinical collaborators and my PIs for this wonderful work, and I can actually accept questions because I think this is the end of the session. Thank you.